I am Jake Eberts. Actually, Rory, do you just start with any sort of comments or anything, or are we good to go?
Oh, do we you buy first?
Dr.
Okay.
I don't know, that feels pretentious to me.
No.
Appreciate it. But yeah, so hi, I'm Jake. So I'm going to talk to you today about getting hepatitis and why it's actually feasible potentially, and you should totally consider doing it. We'll start with kind of a rough synopsis of how this is going to go. It's going to be basically the core thesis, and what you're going to learn from this is that by giving healthy adult volunteers hepatitis C, which is a potentially fatal viral liver disease, you can, having that infection for several months, you can pretty radically accelerate the development pathway for a vaccine for hepatitis C, which is really sorely needed. Some questions you might have are like, what the hell? And if you done ethically, how is that even legal? And like, why the hell would I want to do that? All these are very valid questions. I'm going to try to answer them over the course of the next 45 minutes or so. I'll be talking at you. And like the core kind of like what is connected to Toronto is that there's going to be a forerunner study for this, most likely in Toronto. I'll get more into disclaimers in a minute, but this is still under regulatory review. And as with any medical study, and particularly when it involves something as ethically complex, it's like deliberately giving a healthy person a potentially deadly disease, there's going to be a lot of checks and a lot of oversight. And so what I say here is going to be kind of informed by both open source information that's been published and discussed at conferences, in addition to some of the meetings we've had and the way that my organization has been involved. But things are subject to change. It's kind of the shot there. So yeah, structure-wise, this is going to be me talking to you for like 40 minutes or so. And we're going to start like an introduction to vaccine development and this sort of like a vaccine development. We do have quicker. So me telling you basic introduction, like who am I, what is one DCR? And then the kind of vaccine development 101 we're going to go through and like where a human challenge trial, which is this very specific type of infectious disease trial, fits in. And then talking about hepatitis C a little bit, like what's going to happen in Toronto. And then kind of like why you would want to do this, why anyone would want to do this, and what the particular like EA angle sort of is. And I'll have questions on this, and then we're going to do office hours in the room that starts with the W. So one day here. Hi. So we're a US-based nonprofit. We have a branch in the United Kingdom and Zambia. We lead campaigns in science, policy, ethics to stop the world's most harmful infectious diseases. One of the core sort of advocacy areas that we deal with is people who are in or who want to be in what we kind of term high impact medical research. And one of those is particularly human challenge trials. So these are also called CHIMs, controlled human infection models. There's actually 15 words for them, and it's kind of like an XKCD comment where like every 5 years someone comes to a new standard term, and then it's like, oh, there's 16 terms now. So if I use the interchangeably, that's why. Challenge is like a really funny euphemism. It's kind of like when I stabbed you and was like, I just challenged you as a knight. It means to deliberately infect or expose someone to the pathogen that you're studying. So you'll hear like mouse challenge is a frequently used in preclinical research. And it can be done in humans. One day Sooner began as a grassroots effort at like literally a Google Doc form of people willing to join COVID-19 human challenge trials. That's like a super interesting separate topic. I would love to talk about that office hours. Like COVID-19 challenge trials were kind of at the cutting edge in the sense that like they were both really advanced, but also like very controversial because COVID-19 was an emergent pathogen for which we did not have a treatment and did not know much about. So we'd love to talk about that as well if you're curious. But one of the many disclaimers you're about to hear, we are not doctors. We are not like the wet lab scientists who run research studies. Okay, we are an advocacy group. We're a support group. We biologists on our team. That we don't officially represent any study here. We're like an auxiliary group that's worked closely with some of these teams in the United Kingdom and Canada. But again, nothing I say here is like word of law. So who's this guy? Hi, Jake. I'm the comms director. Weird career path so far. Going to these events is always really funny because people ask like, I'm about to get my master's of public health. Like, what do I do to work in fire security or whatever? And it's like, I had no idea because I got this job by like getting diarrhea on purpose. I joined a human challenge study for Shigella, which causes dysentery. And I like live tweeted like a maniac and went viral. And so they did a BuzzFeed article on there, did some podcasts. I was on the front page of Reddit, which was super weird. And that's how Wendy's here found me. And I used to work in like foreign affairs and like US-China policy stuff, but completely separate. And then I got Zika later. Wendy's here does not require people to get like exotic diseases as part of like employment. That's like, that'd be bad even for like the lax standards of the U.S. employment law. But yeah, you can imagine it kind of attracts a certain type of person. So I actually didn't get Zika. I was challenged with a sham, with a placebo. So all the valor and none of the problems. But yeah, so okay, it's a fun talk. Also, we're going to get to actual unit. But first of all, it's not medical advice. It is not officially approved by a state or research team or institution. We are a third-party group. I am not a doctor. I'm not nearly smart enough to be a doctor. As we're talking like EA stuff, no outcomes are ever guaranteed in medical research. Okay, this is not like kidney donation where you can be reasonably sure that you give a kidney and like some marginal person's life is safe. Okay, vaccine research is speculative, it is hard, and more often than not it fails, right? So we're going to talk about modeling of like what impact looks like in terms of DALYs and stuff like that. But you should not enter any sort of medical trial thinking like, okay, like this is going to linearly lead to a vaccine down the road. You're not a bad person if you hear this and you're like, wow, that sounds really insane, I don't want to do it. That's actually pretty normal. Intensive medical research is like probably not good for most people, is not suitable for most people, right? And that's totally fine. And neither I nor my employers want to be seen or have any sort of like direct financial interest in the outcome of the studies we talk about, but that does not mean there's not a conflict of interest. Okay, so we still have one that says that we are funded or to like promote these sort of studies generally. And as I just mentioned, we work really closely with the teams that are kind of proposing these, and like we have a professional and like reputational interest in like getting them across the finish line. So like keep that in mind as I'm talking about this, because I might have an overly rosy view of like how this stuff works. Okay, fun stuff. All that stuff's boring. Vaccine testing and human challenge stuff. Simple crash introduction. You're a little lab coat scientist. You have a vaccine. What do you do with it? The first is you put it into a bunch of like little experiments with mice and then do some stuff in vitro. And you're trying to answer fundamental questions like, does it look like it may work, like it might work? And like, does it look like it's going to cause problems for like these mice and like maybe people down the road? If the answer is no, you can, if you get some funding and commit someone that's worth the time, this is for drugs and vaccines, but we're going to talk about vaccines mostly. You go to phase 1, which is first in human. That is going to look like typically for a vaccine, giving volunteers increasingly escalating doses of the vaccine over time, often many times in excess of what you would actually give them if a vaccine were approved. And you're answering those kind of same questions again. Like, does it look like it might work? Maybe. And for the most part, you're really concerned about safety in phase 1. Then you do phase 2. So the classic phase 2, it depends on the disease. And as I mentioned, this is kind of a simplification, but phase 2 is trying to figure out both signs of efficacy and of safety. And that's, you can do both perfectly when you're just doing a trial on a couple hundred people. So in the phase 1, you're talking a couple dozen usually. Phase 2, we're talking maybe of a couple hundred. And that again depends massively on the disease you're talking about. But safe to assume it's a couple hundred, maybe 1,000 or 2. And so classic randomized control trial, you take the vaccine that you've created in the lab and you give placebo, you give them to people, you wait for people to turn yellow. And like then you run some fancy statistics on like, did the vaccine prevent people from turning yellow, like getting infected with like whatever you're concerned with? That can take a long time depending on the disease that you're studying. And if that works, then you go to phase 3, that you can raise a crap ton of money and get people to care. You can do that, but like with 100 times more people. Some phase 3s for some diseases have like tens of thousands of participants across multiple countries. And if that works out, you will have a better sense of like the safety profile and the efficacy. Typically, regulators like Health Canada and the FDA in the US want to see like rule of thumb is like 2 independent, well-designed phase 3s before they want to consider something for licensure. But that's, again, huge generalization, and it's very fun. But then you get approval for US Health phase 4, like post-market surveillance, where you're like confirming that it's in fact safe and effective. Yeah, so human challenge trial usually comes in phase 1 or 2. As opposed to a phase 2 where you have like hundreds and hundreds of people, you have a very small number of people relative to like what you would otherwise be doing. You have the same kind of setup, but instead of waiting for people to get infected, you just like give them the disease, and then they turn yellow, and you've done it much more correctly. And there are a couple of other like downstream benefits of that, right? One is with you just get a lot of granular data. Like when I was given shigellosis, dysentery, every time I pooped for like 9 days, I had to do it in a little special bucket and bring it to a nurse, and they granularly analyzed it, right? Which is gross. But that gave a lot of information that you just cannot get in any other circumstance. Malaria, which is one of the most well-characterized diseases in terms of human challenge. Basically, every malaria intervention in the past 50 years has gone through human challenge at some point. We've learned a lot about malaria and the way that the malaria parasite interacts with the human body, and the way it interacts with treatments and stuff just through human challenge alone, because you just get so much data. And it's usually a human challenge will like be used what we call down selection. So if you have a bunch of vaccine candidates, especially for diseases that are not particularly well resourced, like diseases of poverty, basically like hepatitis C, for example, or malaria, you probably don't have a lot of pharma money to throw out of them. And so you're trying to think of a way that's relatively efficient and relatively quick to select the vaccine candidates that have the most promise. And that's really hard because, again, even a phase 2 is often very, very expensive. And so doing a human challenge study can like really, really rapidly accelerate that pathway. And like there are kind of clear scientific benefits too. So that's kind of the basic case for a human challenge trial for a vaccine. And here's kind of again a kind of visual kind of comparison. You just do sort of the same thing with a lot fewer people. human challenge studies actually go back to like Edward Jenner, the guy who invented the smallpox vaccine. He did something that isn't known out today, which is challenge a child. He variolated the child, and the child survived when they were challenged with smallpox. That's bad, obviously, but we don't do that. Since the 1980s, the world has gotten much better about like not doing horrible things in medical research, to put it lightly. And so this is from a systematic review that one New Zealand sponsored, was peer-reviewed and readily published in Clinical Infectious Diseases, which is a pretty reputable journal. And it just shows kind of breakdown. If you can't read them very easily, like some of the most common pathogens historically for human challenges, the EAs have been Plasmodium, which is the malaria parasites, influenza, rhinovirus, E. coli, Shigella, which I got RSV, the most recent Pfizer RSV vaccine went through a human challenge. And more recently, we have ones like COVID and upcoming ones like hepatitis C. Overall, with this systematic review down to, it's like about 15,000 people have been like challenged since the 80s. And in studies carried here, there have been 0 deaths. And about 24 what are called severe adverse events, namely the unexpected ones. So things like hospitalization. It's just like a really strong safety profile. And past returns do not predict future results, especially when you're talking about separate diseases. But that does speak to the degree to which like safety is really a foremost consideration for this sort of research. And here's kind of like some wins list. The R21 and RTSS malaria vaccines both had extensive human challenge with COVID. One of my coworkers got malaria on purpose while he was a grad student at Oxford as part of like the adjuvant selection process for R21, where they chose the kind of thing that makes your body react to the vaccine more. In non-vaccine, the Jynneos vaccine for smallpox/mpox was interestingly actually done by challenge. You can't challenge with smallpox directly, obviously. But you can use the attenuated virus, the attenuated vaccine, the ACAM2000, and do a proxy challenge. They also did that for tuberculosis using the BCG vaccine, because that is technically that's still an active pathogen. Multiple type of cholera vaccines, and then like importantly, lots of failures. Like the whole point of this is down selection, so you can quickly identify failures in vaccine candidates that just don't work, right? And that's great, and that saves time and money and allows resources to be conserved and like put towards other more promising candidates. So really run through this quickly, what this looks like in practice. You recruit a bunch of volunteers, you randomly assign them to a placebo or vaccine group, and then sometime later, you give them that stared sample of the pathogen. In my case, for Shigella, we had the experimental vaccinations, and then 2 months later, we went to inpatient for this vaccination stuff. And so that's where they kept us and monitored us. And so for some, like Shigella is really highly infectious, so that is where you would stay inpatient. For malaria, interestingly enough, they're often outpatient because we know how malaria, like it takes malaria a certain amount of time to basically gestate within the liver after injection, and you just do whatever you want until that like 6th or 7th day, at which point you come in every morning for blood smears. And like the second that they detect the parasite in your blood, you come in and they just do treatment. Super fascinating. And so then you do treatment, monitoring, so on and so forth. And then you run a stat and see, does it look like it worked? The human challenge models are pretty cool. They're not perfect though. There are a couple reasons for that. First, they're still pretty weakly powered. You only have a couple dozen people, or maybe a couple hundred at most for like larger models. And you can't do the pathogens that might cause serious long-term harm, like are likely to, or even sort of maybe likely to. HIV, anything from the Ebola virus family. And up until the past 10 years, hepatitis C fell into this category. And there are lots of technical limitations, like they're not appropriate or suitable in this stuff for all pathogens, even if you could theoretically do them ethically. And they take facilities expertise, right? You need like often quarantine facilities and you need infectious disease research facilities that can handle this. And it takes time to establish these samples of the challenge agent or this infectious dose, which we're going to talk about in a second, because that's going to be really important for this Toronto study and they keep foreshadowing. So the other question is like, why do people do this? And the two most common factors in the literature and what we've seen on what you've seen here is like money and altruism. I don't think this is mutually exclusive. That's why I got dysentery. I kind of joke with my friends, like I can be self-righteous and be like, I saved the world and also get paid like $7,000. Like it was a win-win. And so in the EA angle, I would say like the challenge studies are often a bottleneck, right? It's, as you can imagine, it's kind of hard to get people to sign up to get like dysentery on purpose. And so high quality volunteers who are really dedicated and who understand what's being asked of them and are willing to kind of like really make changes to life plans for a couple months to participate are very valuable. Yeah, it's like kind of hard to come with preclinical candidates for vaccines, but getting that jump from phase 2 to phase 3 or even phase 1 to phase 2 is pretty difficult. And that's why challenge models can be so valuable. And I think it really is a question of like triaging binary resources. That's kind of like the core of what a challenge study is all about. Like you have you only have so many resources to test vaccines, and particularly again for diseases that do not receive like the amount of resources relative to the actual burden they cause globally. A challenge model can be very helpful in down selection of vaccine candidates. So with all that out of the way, we're going to talk about hepatitis C and see the challenge studies that are proposed in Toronto. Background of Hep C. It is a blood-borne viral disease of the liver caused by HCV, the hepatitis C virus. It's a gradual and chronic disease. So it usually stays someone, they don't clear on their own for basically the rest of their lives. It's over the course of decades. It will cause cirrhosis, damage to the liver, which can lead to cirrhosis, which is like really serious scarring on the liver that interferes with its capacity to do its job. And that can also lead to things like hepatocellular carcinoma, which is liver cancer. That's usually how hepatitis C kills. About 50 to 60 million people are infected with hepatitis C worldwide. Most of them don't know it. And depending on the US, the WHO thinks about 250,000 people die every year because of it. US CDC estimates ever in the Global Disease Report in the Lancet estimates closer to like 500,000. It is otherwise mostly asymptomatic though until it's too late. So you can live with it for decades. It won't be clear until you go to the doctor and you have advanced liver failure. The exception is a minority of newly infected people when they get an acute infection, which that first 6 months do experience like general sort of like illness. So flu-like jaundice is the one that's a little unique. It's basically turning yellow, but otherwise painless, the jaundice part at least, for intermittently for a couple weeks. This is not well characterized in part because acute infection is so like generally considered to be pretty mild, and most people don't realize they have it. And unless you're like actively turning yellow, like people don't, like they might think they have a cold, like the cold or something, and then just kind of like over the course of a week or two, just like walk it off. So we don't really know that much about like the specifics of acute infection, like how many weeks might it go on? Be like 2 or 12, unclear. Which is kind of testament to kind of how little attention received, but because it is not particularly severe. About 1 in 3 people, what's interesting about this virus, about 1 in 3 people spontaneously for reasons we don't quite fully understand, can fight it off on their own. It's actually those people who are most likely to show symptoms. So you have really bad symptoms, like in the acute phase, like you're really feeling bad, like fatigued, and you're super yellow, like Homer Simpson. That probably means you're doing a really, really good job of fighting it off on your own. It's like Mothafov, but it's less fun for the individual involved. But there are people who have dermal immunity, and particularly you see this among people who inject drugs, injecting drug using populations, people who have reliably fought off infection 4 or 5 or 6 times in a row. And that's interesting because that does mean that like you can, in fact, like that is an indicator that immunity is possible in the human body. Which is not true things like HIV, at least. Yeah. And like I said, in 2014, up until 2014, you had these therapies called interferon therapies, which were really, really unpleasant. They had a very low, they had like a 50% cure rate, really generally just considered kind of suck, and were kind of a crapshoot. In 2014, the FDA approved these classes of drug called DAAs. I've heard the term miracle cures around by hepatologists and gastroenterologists before, and it's like not a term they like use lightly. They are like really a game changer. It is now very easy to cure hepatitis C. That the 95 to 99% rate is really impressive because that includes real world populations, and in particularly site populations. You're talking about people who inject drugs who often have co-infection with HIV with hepatitis B. We're generally like their immune systems are not in tip-top shape. So even among those populations, and even among populations like that for whom adherence to treatment is pretty low, you have really, really high treatment success rates. But as rare as that is, DAA treatment alone is not enough to like stem the tide of HCV. A lot of modeling suggests that HCV is going to be pretty unique, and now it's one of the diseases that over the course of the next several decades, in absolute terms, is still going to increase, as opposed to things like malaria and tuberculosis and HIV, which like we're generally expecting decreases in total infections. So part of that's because there's no vaccine. The reason there's no vaccine, among many, is like, for one, you have the background political conditions. This is a disease that affects people who are typically the most vulnerable and marginalized in society. In the United States, that's injection drug users. They're not a politically sympathetic group, right? Unfortunately, politicians and society just does not care that they are infected with this disease. And commercially, it's just not particularly attractive. And the same thing goes for the vast majority of people who have this are in the developing world. It is not a particularly attractive target, and is not something that like voters in liberal democracies in the West care a lot about. So with that, there's also kind of the mechanics of hepatitis C. So it's blood-borne transmission. That means you have to contact, like infected blood has to have contact with your blood in order for that, for transmission to occur. And that means it's really unpredictable. So like malaria or the flu, where you can like pretty reliably assume and it's going to happen, in a wave, of course, if I know November, October, whatever. You have to, you basically have to imagine giving this to like thousands and thousands of people in previously like Bangladesh, for instance, and like to hope that at some point there's a hospital that doesn't sterilize its equipment properly, or a tattoo parlor, and you maybe can try to find those people who got hepatitis C from it. It's like it is so logistically infeasible that it just cannot happen, basically. So you have to do trials on people who inject drugs. And that's the roughly hypothesis, like 1 in 12 people who inject drugs in any given year are going to probably contract hepatitis C. And as you can imagine, doing a vaccine trial among people who inject drugs is really, really hard. It's ethically complex. It is slow. It is very difficult. And it's not fun, to put it bluntly. There is you can see Andrea Cox, who's a professor at Johns Hopkins, who ran one of the sites that did the only completed phase 2 among people who inject drugs to date. This was for the CHAD and SME, NBA and SME vaccine trials. It took 6 years, and they recruited over 500 people who inject drugs, did the randomization, and as is often the case for studies like this, did harm reduction measures. And so they would like walk people from their clinic to needle exchanges. And so it's kind of like unfortunate kind of reversal, or this unfortunate kind of catch-22, where they were trying to do this, but every time they failed and someone they knew in the study got infected, that was like what the study needed to power itself. And that was really not fun. And that's like not fun as a clinician, and people don't want to do that. So as a result, there have been no phase 2s for HCV vaccines since this one happened and failed. Part of that's because of COVID, but to our knowledge, there are no others that are planned. Like, it's just not going to happen anytime soon. So that's why we had to be movement for human challenge studies for hepatitis C. This is a joint statement that Wendy Sear helped put together and got published in the Lancet Gastroenterology Hepatology. Now that we can treat and reliably cure hepatitis C, like the case is much stronger. And so 121 signatories, 120 of them plus me. Everyone besides me was like much more qualified. I'm really, really proud of bagging Harvey Alter and Charles Rice. These guys won the Nobel Prize for discovery of the hepatitis C virus in 1989. So like they're the ones who like found out that it exists. And a key quote from this is like the impact of the vaccine itself for hepatitis C would be enormous. You would reduce transmission into red cirrhosis and most importantly, reduce the rate of liver cancer. And so there's a pretty strong consensus among hepatologists, virologists, that this is something that can in fact be done. If we want a vaccine, it's going to need to happen. So then the fun question is like, so what does that actually mean though? Like, how do we know how much impact it's going to have? You can, posit how much a vaccine would reduce suffering in terms of like disability adjusted life years. But like, what does that mean if you want to actually participate in a human challenge trial? And very rarely do you have kind of like anything giving you that sort of sense. This is a paper, it's a pretty simple model that was published in Clinical Infectious Diseases by Alyssa Belinsky and several, is a professor of public health at Brown, and several other researchers. And they just sort of set out a bunch of variables as to like what sorts of things will impact the ultimate, like what sort of things kind of cash out into impact for a hepatitis C vaccine. Some baseline assumptions that there's a QR code at the end where you can like do a really fun little like web model of this where you can like play with the variables. But with pretty conservative baseline assumptions, we're talking like they're like assume that the challenge model is used for 3 vaccine candidates only, and each one has 11% chance of success. That's roughly how many like the percent chance that like a phase 2 candidate ultimately makes it to the end. It's like pretty low. If you assume that each challenge trial is like 5 years shorter than like the corresponding field trial, and that a vaccine would be 70% effective or be taken by like 23% of the population that ultimately needs it, you get about every future infection, like by 2050 or so, you get like 828 infections of variant with hepatitis C for every single person who's given hepatitis C as part of a human challenge study. Modeling is hard, and there are lots of reasons to be skeptical of this model. So one is that it's just too liberal optimistic. There are reasons for that, like hepatitis C could just be really, really hard to vaccinate, right? I mentioned how hepatitis C, for some people, you can generate durable immunity. We don't quite understand why, but that doesn't mean that necessarily everyone will. And it could end up being like HIV, where we try for decades and no vaccine occurs. It may be the case that as direct-acting antiviral prices fall and become more generic, that it just becomes much more cost-effective to do these massive test campaigns. Some countries have done this very well. Egypt is a good example. Egypt as a result of earlier screening and like public health interventions, accidentally infected a lot of people with hepatitis C before the virus was known and could be tested for in the 70s and 80s. And they did a great job of like negotiating with drug producers to create a national campaign that has massively brought down the incidence of the disease. But that took a lot of state capacity and was a particular focus of Mohamed Morsi in Egypt. And challenge trials may still be really slow. A lot of it's going to depend on like how many people clear the virus naturally on their own. Basically, if like 90% of people clear the virus on their own human challenge trials for some weird reason, that means you have to have a pretty massive group to be able to detect statistical significance for that vaccine to work. And so we don't know what that rate will be. It's usually about 1 in 3, depending on the genotype of the virus. We talk about in a minute. That could be a reason that the model just like fails. And in general, vaccine developments like just really are. There are also reasons that this model could be really conservative. For one, they assume that the choice is between human challenge trials and like traditional trials in general. But what we've seen, again, we're aware of no phase 2 that's been proposed anywhere in the world. So the question is not so much like, Is it going to be faster than a challenge trial? Is a challenge trial to be faster than like phase 2 development, a traditional appeal trial? The question is like, it's just like there's nothing nothing that happens besides a challenge trial. Like there's just not anything in the foreseeable future that's going to occur, because it is so difficult to do in phase 2. Also, to our knowledge, we do think there are a lot more candidates out there than just 3. Now that does not mean that every single one will be tested. And it's sort of if you build it, they will come. If you are like a promising young researcher or an academic and you want to design a vaccine, you're like looking through a menu one day or like texting your friends. Hepatitis C is not an attractive one because you will probably learn very quickly, you don't already know, that it's like you'll have wasted your time because you'll come up with a very nice phase 1 candidate if you're lucky, and it's just going to sit there. But if you know there's a human challenge model available, that creates a lot more, that creates a pathway for you. And so that becomes much easier, whether you're a pharmaceutical company or a researcher. So that's another reason to believe that there'll be more vaccine candidates that are tested. And you see with things like the hepatitis C, the B vaccine, that even in really, really under-resourced areas like sub-Saharan Africa, you have really high uptake, like 70% higher. The Hep B vaccine is actually astonishingly effective. It's like 90% effective. And so even under some of these assumptions, Hep C is probably not going to get there. But there's still a lot of reasons that you could think that 1 to 824 infection ratio is conservative. I will know that that model does not include the initial trials that are going to happen, hopefully Toronto, and require establishing an inoculum, which I'll talk about in a minute. Which is like about right now, actually. I don't know my own presentation. Okay, another final set of disclaimers. Case over Mills only. I am not a what? I'm not a doctor, and nothing is final until it receives regulatory or research approval. Research methods over approval. If we didn't believe this was more likely than not, I wouldn't have hauled myself here from DC to not that Toronto's bad or anything, but like I would have seen, if I had we thought this was not like something that's at least possibly going to happen. But we are again not speaking on behalf of any study or any institutions that will run them if they are approved. A lot of this information is drawn from public research papers and conference presentations and our discussions with investigators, but. If these studies are approved, you would go through a really intensive informed consent process that would involve things like quizzes and stuff. And so don't like hang on every word that I'm saying here. This will be a long process if these are approved and if you want to join. So what does it mean to establish the inocula? Basically, you can't just like pick up the pathogen off the streets and just like use it for fun. That's probably that. You have to like characterize it. Challenge studies have a very, very specific sort of a requirement for the agents that they use, generally speaking. So when I was infected with dysentery, it was not just like they found somebody with really bad diarrhia, like in a hospital, and just like I took that. It is a strain that has been like really well characterized for like decades, and they know is not resistant to antibiotics. That basically there's a lot of data showing like this is what happens when you give this to people, right? Down to like the attack rate, which is the rate at which people get actually infected. So we all took the exact same basically shot class in this bacteria in solution, which was super fun. And about 80% of us got sick, which is right what you expect. Some people take the exact same dose and just don't get sick at all. The human body's weird. So for HCV, you want a vaccine that protects against most genotypes, which are kind of like strains. And that means that you're going to need a sample of an inoculum for each of these genotypes of the virus. And so in the initial study proposed here at Toronto, you will probably, that's the goal, is like to basically get this infectious sample. And get a large amount of it so that it can be used in future challenge trials across the world. So if an investigator in France or the United Kingdom wants to do this human challenge study, they can use the exact same inoculum that other people around the world are also using. So you can really quick direct comparison of vaccines as publications arise. The basic approach here in Toronto, or the proposed approach, is like you're going to really start small with like 3 or 4 people, one for each genotype, depending on if you can get this genotype. What you're going to do is you're going to find people in the general population who have hepatitis C naturally. And that's going to probably come through Canadian Blood Services or or might come through Canadian Blood Services or other like institutions in the area. You're going to basically track these people down and say, hey, we know you have hepatitis C of this genotype. They're going to explain this concept to you and ask if they can basically get your blood. And then they're going to use that sample to be able to like treat that donor for free. They make sure that person has been treated successfully. And then they're going to run some very fancy, very like expensive tests to sequence the genotype of the genome of this virus to make sure it doesn't have things that are known to be associated, like are associated with resistance to DAAs, those treatments I was talking about. The reason you want to do this is because when someone has a chronic infection, they're much like the viremia. The level of virus in their blood is much lower than when someone first has that infection, which means you have to get a lot more blood to be used to like get the like samples you need to infect people in the future. And that means you have to like space out collection of blood over a pretty long period of time. And the tricky thing about HCV is it is very prone to mutation. And that means you would probably then have like quote, what happens is you get the evolution of things called quasi-species. And so you would, if you do a chronic donor, that means you're like getting basically a little bit of a different virus each time, and you don't want that. So tube donors are one of the reasons that you're going to be infecting these people first for the chronic donors inoculum, and then going from there. And then you're going to basically take those inoculums. So basically what you do is you have this acute donor who then has generated like highly infectious blood samples, and then you use that to kind of characterize the infection. So for genotype 1A, you want to see how many people roughly are going to clear the virus on their own with that genotype. Isn't 20%, it's 30%. Then you do that 4 times. And that also helps figure out Hepatitis probe, like confirming that you can, in fact, deliberately infect someone with viral hepatitis, and it's fine. Like I said, this is what you get for not understanding your own PowerPoint, but this is kind of the basic run through, right? So we're going to start with getting the sample for free, screening the sample, and then getting to a very small number of volunteers based on the genotype, monitoring them. Then you're going to collect these little samples from them. That's going to involve a transfusion process, which is like pretty intensive. Then you get them treatment and continue monitoring, and then you use those samples that are collected as part of a biobank, and there's going to be like a committee probably that decides like who gets it at what time, like what vaccine candidates they're going to be tested first. But that's the general sort of gist. So basically, like whoever's in these early stages of these studies are like very, very directly involved, viscerally so, in like any sort of subsequent vaccine testing for hepatitis C vaccines. If they would argue that I presented to you is correct, that it is in fact necessary to do hepatitis C human challenge trials to do vaccine development. So in terms of actually participating in this, the RUP timeline for in general would be about 12 weeks of infection, followed by the 8-week treatments. The DA treatments are like a daily pill. They're really well tolerated. It's just like a pill you take. And then you wait for what's called sustained optimal response, SDR, which is basically just the benchmark to show that like, okay, it's out of your blood, you're cured. Congratulations. In terms of like the mechanics of it, again, a lot of this is subject to change, to discussion and stuff like that. But it is going to be pathogen rather than, and it's going to be pretty heavy on appointments. So, and those are things like blood draws, health assessments, questionnaires, some other procedures. And that's going to be really intense in those first couple weeks and months after infection. We're going to start spacing out pretty gradually. Free treatment is free. DA treatment in the United States costs like $47,000 for a single course. Of course it does in the United States, but it's also still pretty expensive in Canada. That would obviously be something you put on a participant. And you would have the right to withdraw at any time. That's true of any even job study, which actually raises some very interesting bioethics questions about like, if I want to withdraw with actively infectious disease that could cause serious harm to like society, what do you do with that? That's a fun question. But you do the right to withdraw and receive treatment for free still. And like all studies, there's like compensation for this. I can't get too much into detail with that. I will say one new senior has pushed for relatively high compensation, given that you will be living with a stigmatized infectious disease for several months. But to give you a range, when I got dysentery, it was like $7,000 US. The highest I've seen for a human challenge study was about 10,000 pounds. You can then for a malaria challenge that required like 93 visits and like reinfection with malaria on like multiple occasions. So that's like the highest I've seen. What are the risks? Long-term risks. And the serious one, of course, is liver damage, right? Liver chronic hepatitis C can and does cause damage to your liver. It's bad for you. The question is, does it cause serious harm to you in the short term? And there's some really interesting ways you can figure out the answer to that question, particularly if you go back and look at cohorts that have previously been infected with hepatitis C accidentally before we discovered the hepatitis C virus. In particular, this is Germany anti-TE cohort. Anti-TE is part of the blood part that's given to people who are pregnant at some plants. And East Germany in the 80s, they gave this to hundreds and hundreds of pregnant women and a lot of them got hepatitis C. But a fraction of them clear the virus on their own. And if you then, like otherwise healthy people, like you then go and examine like their health outcomes over the next several decades, you can see that there's no significant difference in terms of like actual like liver cancer or liver failure or anything like that. There are some cohorts where that's not true. Taiwan Reveal and U.S. Veterans Affairs do show differences. That is probably because they don't control for hepatitis B co-infection, which is really common, and or alcoholism, which is hard to do. And so like the only clear set we have are from these anti-D cohorts in Ireland, Germany. You can see Xu et al. Here at the bottom do a pretty good job like breaking down that evidence. And it'll also be up to regulators. Like we found this argument pretty persuasive one day sooner. Regulators sound like they might agree. And so if they do, like there would be, it's pretty strongly clear that, acute infection is not something that's going to cause lasting health consequences. The one thing that will not go away though is you will probably remain hepatitis C antibody positive for the rest of your life. And hepatitis C is a stigmatized disease. And in Canada and the United States, it is associated with injection drug use. So you will not be able to donate blood. And there's a world in which maybe you have to go to the emergency room and someone sees your HCV positive status, HCV antibody positive status, that makes assumptions about you or like your lifestyle that are unfair. And then they treat you differently because of that in a healthcare setting. That is something that's like pretty important to acknowledge. And that's something that like you might get a certificate saying like, I did this on purpose, but that's like another long-term consequence. Short-term risks are, the primary one is risk of systematic acute infection. Like I said, most people don't have symptomatic infections, but if they do, it's usually transient or across the space of several weeks. Rarely serious enough in your daily life, but it could include things like general sense of malaise, fever, fatigue, jaundice, what's the fun one, and then like nausea, abdominal pain, among others. There's an extremely, extremely low risk of what's called fulminant hepatitis, which is basically acute liver failure. That is reversible with DAAs. That's like rare enough to where like it like warrants case studies on a dome when it does happen. Like it's not unheard of, but it's very rare. It's like extremely rare among healthy people. To the point that it's not expected. Like if there was an actual functional risk, if any of the researchers all felt there was like even like a 1% or 0.1% chance that someone gets acute hepatitis, they will probably not be proceeding studies to begin with. But again, that's a question for regulators. And then the side effects of DAA treatment, really well tolerated. Very rarely in the clinical trials for DAAs did you see people withdraw because of intolerance to treatment, but like theoretically possible. It happened every once in a while. So other considerations for this sort of like discussion in early weeks or months, and you're joining us like this, travel would be pretty restricted because they want to keep you in the area to make sure you can come to your check-ins. But like coming to every single one of these check-ins is like critical. That's like something that is a really big problem in early stage clinical research is people just like don't show up sometimes, and that like creates gaps in data and just like makes the general research enterprise bigger than otherwise it would have been. Alcohol use. If you're thinking right now, wow, I could do this, like drink less, like you can still drink, but like whatever drinking you think is appropriate, like do -1 drink and you'll be fine. That's like general good life advice too. He says how to go to the steak store last night and how you like a little too much. So like that's just one that he consider. Another thing that comes up a lot is questions about transmission to others. Because this requires blood to blood contact, you cannot transmit this to other people in day-to-day life. You would have to like share a toothbrush, which like ew, don't do that. Or like use nail clippers that you like cut yourself with and then someone else does it in the same time period. That's just not something that's going to happen. And obviously for studies like this, you're not going to be accepting people who have signs of objection for use because that is really easy to spread it with, right? Microscopic amounts of blood on needles and things like that do in fact lead to infections. It's also technically possible to transmit sexually. That's really, really rare. Is probably likely that just would be on the safe side, like you'd require to bear contraception. As part of an hepatitis C duration of infection. And some general eligibility requirements that we think will probably be there. You should probably, you'll leave out at the age of 40. You can't have an history of alcohol or substance use disorder or like other significant liver damage. And no chronic autoimmune condition or diabetes, including diabetes. And the first few handful of participants are going to be male, and then open up to both the sexes. The reason for that is because of risks associated with transfusion. Pretty rare. This thing called transfusion acute lung injury. Very rare, but more common in females than males, and that's just one of the reasons they're going to like. Again, there's a lot of like risk reduction measures that are in place here, which is why I'm misappropriating the Swiss cheese model because I think it's fun. It's kind of reverse, whereas like the Swiss cheese model, as we all felt traumatic, remember from COVID, is about like preventing infection, whereas this is like, here's why we can infect you. And the answer is because like several measures of safety considerations are in place for studies like this. The first is like they're going to really stack the deck with participants, right? You're going to make sure that the people who join are very healthy. They're like an ultrasound of your liver, basically, and it has to be like super squeaky clean. You cannot have any sort of like history of any sort of like indication of liver illness, anything like that. They're going to sequence, like I said, this HCV sample before you put it into healthy volunteers for the first time and make sure that it does not have any sort of indications of genetic resistance to DAAs. And I'm going to treat that donor first, just like confirm again that like it is susceptible to the same type of DAAs that are going to be used in this study. You're going to be monitored very regularly, lots of blood work, and like if the illness does become severe, you'll be able to either the discretion of the investigator or your own discretion withdraw. And again, free DAA treatment. And like I said, this little footnote here, like I make a little Swiss cheese thing, but the holes in each one of these slices is actually much smaller, like much fewer than like that image implies. So no. So finally, we're to the EA angle here. Kind of think of it like timed DALYs, basically disability adjusted life years awarded. Often for human challenge studies, what is most difficult is like it just takes time. Like you have to be willing to go into appointments, and they're often not very flexible. Like you have to be in on day 17 to study because that's what the protocol that the government legally approved says has to happen. And so the first few people to study in particular are going to be immensely influential. That's for a couple reasons. First, you're going to be studying a ton for future trials. And if all goes well, you can work with one A senior and will help make sure that like these are kind of announced in fanfare, so to speak. And the media loves to cover human challenge studies. And because the cohorts are so small, like if you're the first person infected with like genotype 1A, and you just decide, I don't want to do this anymore. Like I'm bored, but like I usually like decide I don't want to keep going to my check-ins because this is annoying. You could like massively set back the study that like would be like several months that are out on the process. And so it's really kind of fragile and not fault tolerant in those first couple people in particular. And to a lesser extent, it's still pretty significant extent in those initial small groups of post-sexit that can be infected with the inocula. And so in general, one assumer's thesis and something we found to be particularly persuasive is that like enthusiastic participants who are well read on the subject can like make a really significant difference in terms of like both considerations of these studies in the public mind and among regulatory and ethics bodies. Like part of the reason I'm up here today is because I've done this twice for different diseases, and that grants a certain amount of moral status. Like I'm allowed to talk about it in a way like ethics conferences and with the powers that be is sort of epistemic privilege that is maybe a little bit weird to me sometimes, but also like makes sense because I'm just some guy, right? Like I'm not a doctor. But so we're helping you kind of navigate that process and like become truly informed about the sort of like ways that you can engage advocacy in this if you want to. Is another sort of value proposition that one of these brings. And one of the reasons I was super excited to talk to this crowd in particular. And so, yeah, that's about it. You can scan this if you want to. This gives you like a bunch of resources, like the model that I talked about. There's a dedicated supplement in the Clinical Infectious Disease Journal that like gets into ethics issues, safety, things like that. And is also where you would sign up if you want to learn more information. Signing up with us does not obligate you to join a study, like I said, intensive full consent process. But if you put your name into the ring here, like you'll be signed up in our mailing list, and you will like hear about the updates as they occur. And if you're like sitting here thinking, no, I live in Toronto over like, I don't know, like I'm a woman, I can't do this in the first round. You still been signed up, and you'll get our general updates and keep updated with us. And I write a really funny newsletter, so you should totally do that. And yeah, so really quick, we're running through some like really common questions, like what if treatment fails? The answer is you do treatment again. There are multiple classes of DAAs, and basically once someone fails that first round, it gives you a second, like studies indicate that that second round of treatment is also by 95-99% effective. So these are really, really impressive drugs. And again, if their protocols details will be up, like they're still to be settled, but like if you have multiple people who fail treatment the first time, they're probably going to stop the study because that's like, so that would be incredibly unusual and really, really strange. How much will you get paid? I don't know. I can tell you yet. TBD. I think it'd be a lot. That's my personal opinion. I think it should be a lot to reflect the value of charity by this stuff, but no specific numbers have been decided on yet. It'll be up to the research flags board. And what about living in Toronto? You should still sign up. And like sign up for my newsletter at least. That made me happy. These studies are pretty rare in Canada. I will say that. The only other place that they've been done to our knowledge recently is I'm interested Dalhousie. They're in Halifax is of the whooping cough. They're not especially common in UH studies. There are a lot in DC just by chance, but like major US cities like New York, San Francisco, Chicago, LA, basically none. And so there are other high impact medical trials you could do. We do occasionally track at when they see like high impact phase 1 trials, and there's definitely a lot of those going on here. But this is unfortunately the time being, so it's going to be probably Greater Toronto area specific. Recruitment is going to depend on like the timeline for regulatory and like its approval. We are hoping in 2025, and that's like a reasonable assumption. It could be the first half of 2025. It could be the latter half of 2025. And that would mean for like the expanded groups that include both sexes, that would probably be early 2026 at the earliest. And is this the only Hep C child model in the world? No, there's a parallel one being proposed in the United Kingdom. I would love to talk about that in office hours about like which one is not better. Like the comparative advantages of both of them, because there are some pretty like notable differences. But in general, like we think it's good to have two because again, like low fault tolerance otherwise, a low failure tolerance, like the biobank could catch on fire for one of these facilities and that could just like set everything back. And so you want to make sure that like ideally you do have multiple nodule. In this case, there are probably going to be two that are proposed. So yeah, that is why you should get hepatitis C, or at least think about it. Tell your friends, tell your family. Tell them to come talk to me, and if they especially live in like places like DC or Oxford, I can give like a menu of exotic diseases that they can get that pick. That's been super fun. But yeah, so that's why I'm happy to answer questions.
So everyone, you guys can go into the swap card for the events. You go in live interactions. Then there's a chat, questions, polls. You go in questions because that's where we can actually rank them. So if you want to ask a question, go ahead. If you just want to rank the questions because you have a specific one you think is good, you can also just vote.
How's I hand too?
So they get a Hep C?
So legally, so what's really funny about these is that as well as the United States and Canada tax clinical trial income as like real income. So I had to like fill out what's called a 1099 in the United States, like an independent contractor form for doing dysentery, which means that to do it in Canada, I would have to have like a work visa. Honestly, I totally otherwise would. Like, But yeah, I can't because getting paid means that I have to be a worker, which is strange, but the way it works.
How are Canadian regulators? Like, are they more friendly towards challenge trials than other places?
Yeah. So the short answer is uh the caveat here that like I'm not speaking on behalf of my regulatory body. This is very speculative, yada yada. So with what I can say is initial interactions with regulators in Canada have been pretty positive. Often in our experience, I can talk about, for instance, Brazil. I was in Brazil last week actually speaking because they're going to start considering human challenge studies for a different disease there. The like medical and science side on visa in Brazil is very like on board. It's the ex regulators who are really, really worried about it. And there's a lot of reasons for that. But it is something I love talking about like, office hours. But in Canada, we've not seen indications that like it's going to be a hostile process. Both in the United States and Canada, like you guys have to understand that human challenge studies are for, and with the exception of COVID-19, for which was really kind of a more controversial case, like they're pretty fine with that. Like they've been coming out for 50 years. The vast majority of human challenge studies have happened in the United States or United Kingdom. And so regulators there tend to be very, very familiar with that. So we like don't think that will be a problem here. I off the record, he says on video, I have a little bit more worry about like what happens often when institutions that do not let research ethics boards or United States IRBs that are not familiar with human challenge are confronted with this idea the first time. They have the understanding of like what the hell reaction. And some people get a little bit entrenched and get really, really dogmatic about it and just like will kill it off the bat. Like the RIB system and IRB system are not particularly like transparent, and there's not like a lot of like appeal processes involved often, and so that's often a problem that becomes a problem. So that's kind of it's separate sort of regulatory concern.
What about the idea of human challenge trials for potentially like fairly severe virus, but in case of like a global pandemic, let's say, where we knew for certain, let's say, that it was fairly deadly, yet it was kind of thing where we had, let's say, tens of thousands, if not hundreds of thousands, of people dying every month. Yeah. And a human challenge trial could like hugely accelerate that timeline, potentially save millions of lives. It was the state of the world.
That's a very good question. And there's a lot of like ethical and regulatory like constraint about that because we don't need to answer. The WHO right now is currently drafting guidelines on like how you consider human challenge trials in controlled human infection model or controlled infection models or human challenge trials in like emergent public health processes. One of the people Wendy Sooner contracts for this, Racine is sitting here right now and will be helping draft a paper on that position paper that we think kind of gets to the core of that issue. But the short answer is like it's really hard because we often don't know how it will be helpful, and especially in an emerging like large scale outbreak. You might just have enough community transmission of a disease to justify a phase 2. Like, I mean, we were with COVID, we did really, really rapid phase 3. In the space of 3 months across multiple countries, you had high quality phase 3 studies done very quickly for vaccines that ended up being quite effective. And so the short answer is like, that's an unresolved question in bioethics and law. And it's a really interesting one that Wendy Sierra follows pretty closely. But it is, it's the short answer, like, it's really hard. Like, it depends a lot on the pathogen and a lot on like the specifics of a given outbreak. But personally, not seeking organization, I think it is theoretically possible and justifiable, given a certain set of conditions about like what an outbreak looks like.
In case COVID could have been like accelerated by like a month or something like that, even if it was a human challenge trial that was done, because you said 3 months ish for like phase.
So for other movie, COVID was unique because we like put like the entire books of the world into it. And I can talk a little more in office hours, but like the short, it's like I think there are other uses for COVID for a human challenge that would have been interesting outside of like vaccine testing, like controlled natural exposures, where you try to figure out how transmission occurs. So we were kind of dark about that for a long time. Like we all thought that it was fomite spread, right? Like we all had that peer were like disinfecting every single thing we touched, right? That just like is not how COVID spreads. So the world in which like those sort of like early stage, particularly for respiratory diseases, human challenge models that might make it useful, but yeah, that's like a lot more speculative. Yeah.
So I heard some amount of takes during COVID that bioethicists just had bad opinions about. Like, there's a lot of grumbling about bioethicists on challenge trials in general. Do you think that was true? Do you think it was fair? Do you still think this was true?
So I will say this as someone who I think disagrees with many bioethicists on a lot of issues, but like, I should put this. I think there are bioethicists who are really wrong and like think some really dumb things. I think that like any field, there's like a pretty diverse range of opinions. And I think that like I'm a little weary of like casting off the field of bioethics as a like sort of like entirely useless enterprise. I think that what is often really important is to remember the frame of reference for like, especially the United States and North America. The reason bioethics exists is because the United States of America has a horrid history of horrible pieces of people in medical research, particularly Black people. And that was like pretty core to medical research for a huge portion of our history. Some of the earlier human challenges were done on prisoners, right? And so bioethics as a field kind of emerged in response in the 70s, 80s to these really horrible abuses like to Stevie Sipple's experiment. And so I think it's important to understand when you consider the field, the reason the field exists, mainly to like prevent those horrible human rights abuses from occurring again. The kind of small c conservatism that exists makes a lot more sense, and I think it's easier to report charitably. I think it's easier to interact with people who disagree with you as you kind of approach from that good faith standpoint. Because you know some disagreements. There are also very biases who I agree with on everything and things are right. But yeah, so I don't think it's like a question of and there's also cultural difference. In the United Kingdom, some regulators were like much more pro COVID challenges, why they did them there. And that worked great. But in the United States, it's like pretty market difference. And there are reasons for that, and there are reasons for that we don't understand in cultural differences. But like I will see like bioethics as a rule, it's wrong. But there you got places that people go in bioethics were wrong. Is there it happens? I like can't figure out how it works.
Yeah, no, this
Okay.
Your question was asked right on the. I printed it in the chat, and then I was like, "Oh, this is the question." Yes, I didn't notice until now. That's all good.
Yeah.
To develop arguments for, let's say, us to go forward with this. Which arguments, when you're talking to regulators, do they find most persuasive or least persuasive?
So the regulators are often regulators don't necessarily judge like the relative, like the regulator's job often to, and I say this as a non-regulatory specialist, is to assess can this be done legally according to laws of Canada or the United States. So it's not their job necessarily to say like, is this something that should be done? Is this something that can be done based on like the requirements that we have in statute? And so that's a bit, they're often very loath to opine of like, I think that'd be a good idea. Again, like, I think that with, so that's like a legal, like, right, like Health Canada in this case, or like the FDA. So that's like a separate thing. And often in the case of, for instance, US FDA, less current Health Canada, the regulatory like interface there is that the Food and Drug Administration treats challenge agents as a drug, or as a like investigational drug, basically, or like drug in the sense of like vaccine, any sort of biologic. And so you have to regulate it in the exact same way that you would like a novel vaccine candidate. You have to make sure it accords something called like good manufacturing practice, and like a bunch of other stuff. But that's like the main interface that the regulators are concerned with. They're not necessarily asking, is this something that like is wise? That is for what basically the statutory authority in the United States for that comes down to. I'm talking news because I understand a little bit more of my shades. Canada's is fairly similar. The like ethical oversight authorities devolve to FIRVs, which are federally like overseen by a separate agency, and they have the kind of final say. Like the federal law requires the United States that you like have a coherent ethics and oversight process, but that's not the job of the feds per se. And so like the arguments you're making in regulatory meetings are often like, here's why we can do this transfusion like justifiably, like here's how we can like make sure it accords to good manufacturing practice, and like a bunch of other like really minute details that go way over my head. It's not so much the kind of core argument. That argument often will play out in I think ethics oversight courts. Anyways, let's go.
Are there like, because you mentioned actually a different type of human challenge that like you test like infection. Is there like any other kinds of information you get from human challenge trials, like how types and that's
Yeah, there's lots of types. One of the more problem was controlled natural exposure. You see all of it, influenza. We like know shockingly little about how respiratory pathogens are spread, and like COVID kind of revealed that, right? And so you can just do things. This is actually really funny. I think it's like one of the funniest possible like trial designs there is, and that's something I would do in a heartbeat. Imagine you like naturally have the flu, and like you're recruited by a study while you have the flu to come in and like sneeze on a bunch of strangers. And that's like straight up something that they have done before, and you like get to sit in like a room and like play Parcheesi or something with someone you've never met who's like actively like sneezing all over your. I think that's really nice and like useful because we don't really tell them that much about how the flu spreads, which is nuts, right? So they do those at University College Park in Maryland, University of Maryland College Park, and I knew about this and thought I had a cold and was like devastated when I didn't have flu. It was just like a cold because I was like so excited to be the person who gets to come and play like Uno with a bunch of random sprigers and I'm drinking snot on them. So like that's the thing, like these controlled natural exposure experience. And sometimes you will do the sky's the limit really now. For Shigella, sometimes for some diseases you'll see what are called like re-challenge, where you're trying to figure out like, okay, if you want to design a vaccine, right, like what are the baseline like strains or like subspecies of this pathogen that you need to have protection against for like broadest spectrum of coverage. And the way you figure that out sometimes can be just by like giving someone type A of this disease and then like 3 months later giving them type B and seeing if they're protected. And so that's like not for a vaccine directly, but is those kind of re-challenge experiments can also be very useful. I'm trying to think of any others that are like usually drug or vaccine stuff. But yeah, those are like just some of the examples. There's some that get a little more obscure. And again, like what challenge, like you can get pretty creative, like what challenge covers, like that Genius, the Mpox / the smallpox vaccine. Like site is like the authors of that did not call it challenge, probably because it does not have the word smallpox and human challenge in the same sentence. But like the authors of a separate systematic review were like, this was a human challenge study. Like it's weird they didn't say it, but like it was, vaccine by proxy sort of challenge.
That'll be our last one, I think, into that cycle. Yeah.
What's the impact in terms of like the demographic that this human challenge is put on, right? Like I'm thinking about, people in Toronto that are willing to do this. You're probably maybe you work from home, you're of a certain social bracket, and thinking about like the data that's coming out of that and how applicable that is to, someone from a different country who maybe culturally they are they don't trust, let's say, Western medicine and vaccines in general, let's say, right? So we're kind of curious, like, sort of how much weight, like, put on this data itself.
That's a really good question, and the answer is like it depends, and sometimes no. I mean, so yeah, the core thing is like human challenges are not represented, right? And so malaria is a great example of where people who are malaria naive will have a much stronger immune response upon initial infection, and often respond much better to vaccines than people who've been systematically exposed over the course of their lifetime, which is sometimes an argument for doing a human challenge in an endemic setting, which Canada has done. They've been Kenya and Malawi pretty successfully and ethically. The short answer is like it depends, and generally speaking, if you don't think the data is going to be useful, you really don't develop the model. But the ultimate purpose of this is basically the idea is, kind of high level, if this vaccine works well in like adults in Oxford, it worked malaria, but it might work well, yet if we do a phase 2. If it doesn't work well in adults at Oxford, it's just not going to work well in like children or anything like that. So like there is an imperfect sort of like, there occasionally have challenge studies for like travelers vaccines for it's like 1-to-1, and Vaxchora is their cholera vaccine in the United States that's been licensed solely on human challenge. They're free rare because often, so it's like, it's still like it's still a gamble. Like there is no perfect data predicted. And sometimes what often happens in Shigella, for example, what's happened many times is you have something that performs pretty well in human challenge, and then we give it in a target population a non-challenge, which is namely children under 5 in the developing world. It just doesn't work too well because children have weaker immune systems and their immunological just kind of profile is very, very different. So yeah, definitely goes back to this, like these models are not perfect, and that's one of the reasons you do a phase 3 is because you basically really can't prove anything definitively to the standards that medical regulators require without a larger scale study for the most part.
But still, let's just skip phase 1 and phase 2.
You're done in place of phase 1. You usually keep doing this is depending on where these are part of the phase 2 process. So like you don't skip into the phase 2 to different. So phase 2 field study you don't do necessarily, or you go to, but it's complicated. But like yes, these are often in lieu of the traditional phase 2 or in the field, but they're still classified as phase 2 or making a spot. It depends. It's really complicated.
Last time, so thank you.
Thank you.